Hello and welcome back to our Bioscience Python tutorials for non-tech users. So last time we made our alignment tool, and that worked great. We can enter files and we can run that, and our program will recognize those files. So we have an interface that is usable by most scientists. The problem we have now is that we don't have anything to actually align these. So we need to think of a way to actually align these two sequences in an accurate way. And so there's a couple ideas that we could have about how to do that. We could just go loop over this one sequence, and for every position in this sequence, we could compare here and see how far, it matches and maybe give that a score. Or we could go by looping over one sequence and seeing whether the second sequence matches, but both of these approaches don't really work because if we were to loop over one sequence, that would assume that the first sequence is the ground truth. We would assume that that sequence is sort of the correct version and we are aligning against that one, when in reality, both sequences might have gaps, they might have deletions, they might have mutations, and so we can't really give that bias to one sequence over the other. Um, so we're going to take an interesting programming approach, and this is where I hope I really get all our life scientists and our biologists hooked on Python and programming, because we're gonna use this really interesting approach called the Needleman-Wunsch algorithm. And so this was an algorithm developed by Saul B. Needleman and Christian D. Wunsch in 1970. So that's about, if for when most people know, that's when sequencing kind of took off as a technology for biology and microbiology. Um, and it uses this interesting approach of kind of pathfinding between two sequences. So you can see this is a completed algorithm matrix, and it works by kind of backtracing along the highest scoring path that we can create through this matrix. Um, and it'll assign, 1 to a matching sequence and -1 to a mismatch. And you see this and you say, okay, that makes sense sort of for the top left here, but how do we get all the way to -7 up at U? And so I'm going to try and explain this algorithm a little bit better because it uses an approach in computer science called dynamic programming. We're going to break our problem down into lots of small subunits of problems. So I think a better way to describe this would actually be to show you right here. We're going to take our two sequences, and I could just put them right next to each other, and we could see that like A matches, C doesn't match, G doesn't match, that doesn't match, T matches. And so this would be kind of our look at what an alignment would be. But it's easy to kind of see that if we want to align this whole sequence, let's say we theoretically had these first like 4 or 5 A's solved in this second sequence here. So we just theoretically had those alignments solved already. And we had this. And so we already know that these two A's are aligned. And so we're breaking our problem down into a smaller problem of saying, okay, we have these first 5 uh, bases aligned, but we don't have the rest of this. But the interesting part is that we don't have to ever do this alignment again if we know how it works. We can just proceed onward from here and continue kind of going down this. And everything that we've already solved is completely solved. We know this because, if we have part of this kind of we have part of this alignment solved up to some index I in our DNA sequence or in our protein sequence, then we won't have to solve it again. And so this is the approach of dynamic programming. This is where things get kind of interesting, because what we can do is we can break our sequence alignment down into this big matrix and say, okay, if we move diagonally down and to the right in this matrix, it means, okay, I have chosen G and G in this case. If I move again, it means I have chosen C and A as an alignment. If I start up here with zero alignment and I move down one, it means I've chosen G, but I have chosen nothing to align against it. I have no base in this sequence up here that's going to align to my G over here. So, it's pretty easy to see that in this first example, we're going to go down because we have a G and a G. And then we're going to go either right or we're going to go left because, well, we're probably going to go right because we have an AT here and we have an AT here. So we go down, we go over, and then we go here again where we align two A's, and then we go down and to the right again where we align two T's. Um, so this from a visual perspective is a lot easier to see than how computers have to visualize it. So computers are good at math, and so we're going to optimize this so a computer can do this really quickly. Um, so by using this approach, we're going to solve our problem for this space and for this space and for this space and for this space and this. We're going to solve our alignment problem for every single space on this matrix, and we're going to use the previous problems to compute the new score in that matrix. So what this looks like is we initialize our matrix. We say we have zero alignment score here, and we have a -1 mismatch score. So if we move one to the right without matching, that's a -1. Two to the right, -2. Three, four, five, six, and up to 7 gaps. Okay. And so these are going to be our base scores that we're going to use to compute. Okay. Now we take a look at this first problem. We take a look at X. We want to say we'll go down and to the right based on anywhere we can get to this space from that's going to give us the highest score. So the way this is going to work is that we have two G's here, both of which have -1 cost for, this gap cost in our alignment. Or we could move down into the right and get a +1 score because the two G's align in this instance. So we're going to move down into the right in this case because that's going to give us the highest positive score. And then we're going to log where we came from in this matrix, or we're gonna recognize that in some way so that when we backtrace from when we get to this bottom right and we have our full alignment, we're going to be able to path find our way back using the maximum scoring path, and that's going to give us our best possible alignment. So this is all going pretty fast, but you can see the algorithm does just that. And then we have two more boxes to sort out when we've solved all of the surrounding boxes, and we basically just for every one box we can either go down, we can go right, or we can go diagonal. And so this is the approach of dynamic programming where we're using all of our previous solutions to solve more and more and more. And this is a lot more efficient than running the alignment for every possible combination every time. It's just running one alignment up to, or it's really just taking the previous scores and adding one more element onto it because we've broken this problem down into a much, much smaller or a much simpler idea for the computer to compute. So this is great for gene sequence alignment because we can kind of just say if it's a mismatch, it doesn't work. But for proteins, it's a little bit different. What we want to use for proteins is something called the Blossom 62 scoring matrix. And this is going to tell us how bad or how good it is for a certain amino acid switch or consistency between two protein sequences to happen, because some amino acids are much rarer or much more important to protein structure than others. And this is where we get a really, really good sense for protein alignment. So we'll say Blossom 62 scoring matrix. Yeah, I'll just do a TXT here. Blossom 62, wonderful. Yeah, okay. So we're just going to copy this because I don't really care about the rest of this. And the Blossom 62, how it was developed was very interesting, but we don't really have to go into that right now. What we are going to do is we're going to make a new directory, documents, and we're going to input this text file, blossom62.txt. Okay, yeah, whatever, sure. And this is going to be our scoring matrix. So we have all of the files that are going to be necessary to do this. We have our sequence files, which I realize now are DNA sequences, and we probably want protein sequences, so I'll change that a bit later. But this will be fine for now. A, T, C, and G are all amino acids. And then we have our scoring matrix. So, you know, if we have an alanine and another alanine and it's consistent, then we have a 4. If we have a tryptophan and another tryptophan, that's going to be an 11 because tryptophan is a very, very rare and very important residue to protein structure. And if we ever change it, we're going to get this big kind of negative value, except for in the case of something that is similarly charged. So up here, you see, yeah, tryptophan. It's okay with sort of switching like that. So now we're going to go through and we're going to make sure that we can get all this text data into our program. And so we're going to start making our first service. And we're just going to call this helpers. Okay. Yeah, so we have our helpers. We're going to signature it again. So Kayla Ellington, 2020. Great. Great. Okay, and we're going to define a couple things here that are going to be really important. We're going to define FASTA to data, and we're going to input our FASTA file, and we're going to raise not implemented. Okay, and that's going to get our FASTA files into an actual sequence in the computer that we're going to use. And then we're going to define blossom to data and blossom. And that's going to be, our arguments are going to be our file paths. So we should probably say file path, FASTA file path, blossom file path. Okay. And then we're going to define our class, which is going to be our model for this actual sequence alignment where we can run our algorithm. Okay. So we're going to call our class alignment. All right, we don't actually need that. And we're going to define an init. Yep. Is not. Yep, not implemented. Okay, is not implemented. Okay, and this is just it's gonna be a good way of telling us as the developer whether we have created something yet or not, without giving us compiler errors for having empty methods in our class. So we can still run everything even if we haven't implemented it yet. And that's going to be useful for defining these methods and how we use them. So first thing that we're going to do is define run NeedlemanWunsch. Okay, and we're going to have sequence 1 and sequence 2. Okay, that's going to be a public tool that anybody can use on this alignment class. You know, maybe they have their sequence of something else. This is just an interesting algorithm that we think is going to be useful for people. So what we're going to do now is, oh, right, yeah. So we pretty much just need this as our NeedlemanWunsch, and this is going to spit out our scoring matrix. And then we're going to have define get alignment. Okay. And this is going to be for the actual for the actual sequences that we input to alignment. And this is going to raise not implemented. Okay, and then we're going to raise not implemented. Cool. Great, 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 great. And then we're gonna have a define align helper. Okay. Okay, and so this alignment helper is going to be important later when we actually need to get the significance of our alignment. So we need to see whether our alignment is really, really worth anything, because you can kind of align anything you want. That's the point of NeedlemanWunsch is you could throw at any sequence of characters that are amino acids and it will give you an alignment back. But we want to make sure that our alignment means something, and that's going to be very important to know. You know, if a better alignment could exist for that sequence of characters, if there really is a relationship between those two sequences. And of course, you know, as scientists, you should know the importance of statistical significance in our arguments. So first, we're just going to implement these two data sequences right now. So we're going to have our FASTA file path, and we are going to get, we're just going to open it, and we are going to read all of its data in the way that makes sense to our program. So with open of FASTA file path as sequence file. We're just going to get everything right now. So text equals sequence file dot read and dot upper. So we're gonna make everything an uppercase character to decrease confusion here. And then we're going to do text.split at our new line character. So if you look in our FASTA files, we have a new line and then we have our sequence. And what we're going to do is we're going to take the first entry because arrays are indexed at 1, and we're going to get our sequence equals this split at 1. And we're also just going to dot, I believe it's strip of, yeah, we're going to strip our last new line character there just to make sure. And it's Python, so, you know, do it in one line or it's not Python. We're then going to just return sequence. Great. Okay, so let's just go back to our command line tool and make sure that this is working. We're going to say sequence 1 equals, we have to import all of our stuff. This is important. So first we're going to create a new Python file called init, and we're going to not put anything inside of it. This is just going to be init, and this is how Python tells that directories are Python packages instead of just normal directories. So now what we can do from this align.py is we can go from services import helpers. Well, I guess really we want service.helpers import all. Okay. All right. And then what we're going to do is, sorry about that. Right. Yeah, we actually don't even need this services. I was wondering why I was having that weird sort of issue. Result reference to helpers. Okay. Yeah, whatever. It'll be fine. Yeah, it'll be fine. And so we're going to go back and we're going to get our FASTA2 data, and we're going to go back to our align, and we're going to say FASTA2 data args dot sequence file 1. Okay, and then we're going to print sequence 1. Right, just as kind of our check. No module named helpers. Weird. Okay. From helpers import module named helpers. Okay, come on now. Right. So we have resolve reference. It's got data, it's got services, it says data, services. And give that a shot. Yeah, there we go. Okay, we just had to enter the directory to make sure it knew where everything was. Cool. So you can see here that Python read our sequence file and gave us this sequence of characters back, which is our characters on our sequence1 file. So we effectively have them in the program. We're going to do the exact same thing for sequence2 equals fasta2 command of sequence file 2. All right, and now we've got 2 sequence files that we can effectively use for our alignment. Now we have to get this big scoring matrix in, which is very, very important to our actual alignment because it tells us how much it costs to change from one amino acid to another in our alignment. So this is going to be a little bit more complicated. We're going to do blossom to data. So with open blossom file path as blossom file, we are then going to actually get this in kind of an interesting way. We're going to say header equals blossom file dot read line dot upper strip, get rid of some extra spaces, and then dot replace double space with a single space because we have double spaces between all of these letters here, and that's gonna cause us some weird problems. So we're just gonna print our header, and let's see. We want to make sure we're actually calling that. So blossom equals blossom to data. And then we're gonna hard code this in because we're not going to be changing our documents folder documents slash blossom some 62.txt. Great. Okay. Cool. So we now have all of our protein amino acids as a header here. So what we now need to do is get all of these into a scoring matrix where we can enter any 2 amino acids and look up the score between those 2. And the way that's going to look is just in a set or in a dictionary within a dictionary. So we can look up all of these specific values. And so we're just going to go back. And for those of you who don't know what dictionaries are just a way of looking up values with keys instead of indices. And so we're going to be able to type in our specific amino acid combination, and it'll come up with the score that it would take between them. Okay, so our species equals header.split. Okay, and we're going to split this down all the spaces so we get each one of these characters individually, and that's going to be our species, and our score matrix is going to be that. For each line in our blossom file dot read lines, and we've already read one of our lines, so we know all the rest of it is just going to be data. A row is going to be equal to line dot upper dot strip, get rid of the rest of the extra spaces, dot replace our 2 spaces with one space. And then we're going to dot split again at our one space. Cool. Species 2 is going to equal our row at 0. Okay, so what does that mean? That means that as we're reading all of these lines, we want all of the scores, and we want to separate that from the actual entry or the key to get that specific score. So this is our second species is going to be our row in its very first entry. Okay, so our species 2 score equals that, and then for I in range and our length of species, species to score at, we're going to enter new entries at our species at I, because what we're now doing is we're looping across each of these entries and we're logging which of these amino acids are that they're linked to. And so we're going to do our species, our original species, that top row at I equals, turned into an int, our row at I plus 1, because we just got our first entry as our first species, and that's not going to be an integer. Okay, so after we have all that, our score at species 2 is going to equal species 2 score. Okay, great. And now after that, we're done. We can just return a score. So the way this worked is for each line, we're specifying which the second species is, and then we're going to go across the whole header and we're going to catalog all of the scores there. So this is going across the whole header for each of the species going down, and then it looks across for all the scores, and then it just sets our score matrix at that second species. So our score matrix at this is equal to all of the scores here by their amino acid representation, their character. So we return our score matrix. So now let's go back and just try. Let's just try running this and printing our matrix. Print blossom. And this is probably going to look like a giant disaster, but yeah, so it's a giant disaster, but let's just pick a score here to test our view. What looks interesting? This -4 here looks interesting and unique, and that is at the intersection of W and B. Okay, so we go back and we go to our alignment file and we say at W and at B. And this matrix is symmetrical, so it doesn't matter which order we put them in. Yep, -4, perfect. Okay, so that's it for now. We've gotten all of our data into our program, and now we're going to be able to enter it into our alignment and run NeedlemanWunsch on this, because now we have a scoring matrix and 2 sequences. So the exciting part comes next time. All right.